All right. So if the carbohydrate discussion was the most high yield, I would definitely have those pieces down. I would say our discussion about cholesterol synthesis, cholesterol metabolism, as well as some of these fatty acid components would be my next high yield piece of the puzzle for us to learn. So that's what we're going to talk about next. Is we'll start with cholesterol synthesis. So we'll start with page 66 of the PDF, and then I'm going to be going back and forth between also a PowerPoint on cholesterol metabolism, as well as some other fat components as well. So I'm going to share my screen. Maybe. There we go. Awesome. Great. All right. So lipids, fats, and cholesterol. I pumped this out into its own PowerPoint this year because I feel like there is always some confusion, misconception, misunderstanding around cholesterol and its pathway. So first, when we're talking about the cholesterol pathway, we're not necessarily talking about at all the digestion of fats. We're not talking about the breakdown into fatty acids, into triacylglycerols, TAG, or any of those processes. We're literally just focusing first on how do we make cholesterol, which is a really complex, obviously, fatty compound, the backbone of all of our steroid hormones, and important for us to understand. So the cholesterol pathway, the liver is the major site of cholesterol synthesis. So every time we're talking about creation of cholesterol, we're talking about the liver as our main place to go. All the carbons of cholesterol are derived from acetyl group of acetyl-CoA. So if we remember acetyl-CoA, the thing that we created from pyruvate, that exact structure is what we will pull. Instead of going through the Krebs cycle, the TCA cycle, we will use these acetyl groups to create cholesterol from it. And I think there's an example here. Yep, we can see on this page here, our acetyl-CoA structure. We can see it being shunted instead of going through that TCA cycle. We're seeing it being shunted over down this pathway to create our cholesterol. The enzymes for this process are located in both the cytosol as well as the endoplasmic reticulum. Because remember, the endoplasmic reticulum, Golgi body, those are places in cells where cells create proteins and fats. So it makes sense that some of our enzymes will be located in that structure. So this is kind of a big process looking at all the things happening in the liver. We can see our glucose glycolysis process here to pyruvate. We can see pyruvate being shunted and converted into oxaloacetate and acetyl-CoA. We can see some acetyl-CoA being shunted outward, or essentially citrate, and then being converted into acetyl-CoA in the tissue, or peripheral tissue, the cytosol tissue. And then we can see that either moving through this pathway to create fatty acids or this pathway to create cholesterol. So this is kind of where we're at in our process, as we're going to look through this red shunt here. There are 5 stages to cholesterol biosynthesis. So again, we're looking at our acetyl-CoA. This is showing you how many carbons are present. So 2 carbons here. The first stage is we're going to form our mevalonate from our acetyl units. So we can see that step, our mevalonic acid going here. And I'll have another picture later that has the actual full conversion. It might actually be easier if we look at this other one. We'll keep for now, but I'll have them kind of both dueling at the same time. Yeah, this will be easier to see, I think. There. So now you can kind of see it in one nice pretty row. So we can see here, acetyl-CoA plus acetyl-CoA combines together. Our first kind of goal is to form mevalonate here. We can kind of see this process also equating on this side there using HMG-CoA reductase. HMG-CoA reductase, our rate limiting enzyme for this process, which we hear about. This is what statins will block. From there, we want to convert mevalonate into activated isoprene, which we don't see on this page, but you can see on this page here, potentially, isopentyl pyrophosphate. Then you'll add multiple isoprene units together to form squalene, which is a 30 carbon unit. So we can see here squalene, this super duper long carbon unit. You're then going to cycle and create this cyclical structure using squalene as the backbone. So the cyclization of squalene, And then you're going to monitor, or you're going to modify linosterol, which is essentially the precursor to create its final form, which is cholesterol. So 5 major steps. Your main rate controlling step is the conversion to melovinate. If you can get here, you'll likely move forward in this process. So that's why HMG-CoA reductase is our rate limiting enzyme for cholesterol synthesis. Things that upregulate this process that will encourage this process to go forward include insulin, thyroxine, so our thyroid hormone, estrogen, things that will downregulate the opposite, right? Glucagon. Also cholesterol. There's lots of cholesterol in the body. You ideally will not be creating more cholesterol byproducts. Okay. Another way to look at it here, again, this is talking about regulation. Hormones, glucagon inhibits cholesterol formation. Insulin will promote it. It's going to be increased. The conversion to cholesterol will be increased with consumption of carbs and fats because you have more backbones. It'll be decreased with the consumption of cholesterol because you already then will have cholesterol present, or if you are in a fasting state, because this is going to be obviously a lot of steps and it's a high energy process. Okay. So this is occurring again, mostly in the liver cytosol. Okay. This is the process of removing acetyl-CoA. It's just showing kind of these individual bio steps. I really think just knowing that HMG-CoA reductase getting you to melovinate is the key regulating step here. Then when we are actually taking under consideration the breakdown of cholesterol, we need to include our bile salt formation discussion, because that is going to be one of the major ways that we can utilize cholesterol in a form that will start to either be recycled in the form of bile salts or eliminated in stool or feces. So I'm going to walk us through the words first on the left, and then we'll look at a picture kind of these next 2 slides. So I have a picture over on the right-hand side. So when we remove cholesterol from circulation, we are doing so often to create primary bile salts. Primary bile salts are cholic acid plus chenadeoxycholic acid, and they are synthesized from cholesterol in the liver. So we have cholesterol here in the liver. We're going to essentially move this process forward to create these 2 acidic structures that you can see here. These 2 acidic structures are going to be the base or the backbone of our bile salts. Only hepatocytes, liver cells, express this high level of 7-alpha hydroxylase, which is the regulatory enzyme of this process. So this, again, has to occur in the liver. That's why we create bile salts. We create bile in the liver. We store it in the gallbladder. 7-alpha hydroxylase will be your rate limiting step. Okay. Once you have your primary bile salts, they are converted into secondary bile salts via intestinal bacteria, and then secondary bile salts are going to be absorbed from the gut after that formation by bacteria. So it's kind of the cycle. So your primary bile salts are what's released. Your secondary bile salts are formed in the gut lumen, and then they are reabsorbed by your gut bacteria and recycled, essentially. Both primary and secondary bile salts are conjugated in the liver with either glycine or taurine, which you can see here. So these are important amino acids in this kind of cholesterol removal bile acid creation step. They are also both going to be secreted into the gallbladder, into the small intestine with food intake. And both primary and secondary bile salts in the intestine can be conjugated, reabsorbed, and returned to the liver via portal circulation. Liver will then reconjugate bile salts and will secrete all 4 of them into bile again. So your primary and your secondary bile salts can be all secreted from the liver into bile, all secreted from bile into small intestine, and then from there be utilized or recycled. If bound to fat, they will often leave the body in the stool, and that's a way you can eliminate cholesterol from the body. This is the reason why we encourage fiber consumption to help with lowering cholesterol, because we are trying to encourage the binding of our fiber, the binding of our fats to our bile salts, and encourage elimination. We can see bile salts here, secondary bile salts here. How much of this should you know? I would just know kind of the basics, the key enzyme, 7-alpha hydroxylase, and understand that this occurs really in the liver only. Okay. Any questions on that one?
I have a question.
Yes.
So the whole like recycling thing,
Yes.
I just don't really understand. It seems to be just this continual loop. If we're always just reabsorbing them, so the only thing that breaks that loop is getting it bound to fat and
Fat.
removal through the
Correct.
stool.
Through feces, correct.
Yeah, okay.
Yep.
Okay.
So otherwise, this cycle, primary and secondary bile salts being secreted from the liver through the gallbladder into intestines, this will continue to be recycled unless if it's bound to fat and then into feces. And so we can see here that it's like less than 5% actually are being removed in the feces. The majority of them are being recycled, 95%. So it really is not much.
Okay.
Yeah. Yeah. So our body, we really try our hardest to not have to recreate cholesterol. And so anything that cholesterol is converted into, our body tries to recycle it as much as possible because cholesterol is such a high carbon molecule to create. If we start with only 2 carbons and we end up with over 30 carbons, that's a lot of reactions we have to do and a lot of energy input we have to expel to create this one molecule of cholesterol. So once the body creates this, we do our darndest to not have to recreate it again.
And
That's
so
why a lot of these processes are recyclable.
Yeah, and so I guess like our body will even hold on to like a ton of extra cholesterol, and that's where it becomes really problematic because I guess it's too bad because it won't let it go. It just keeps it going.
It doesn't want to let it go. Exactly. Because that if we think about our bodies as why they were created and how they were created, not in the, the state that we are now in society, we were, often a hunter-gatherer fasting state for long periods of time. And so anything that was high energy that we needed to create, our body tried to become really good storage facilities for those items or those ions or those molecules. So we didn't need to actually increase or intake more calories to make those functions work. Yeah. So our body's really good at increasing cholesterol levels because we find the value of it in that it's the backbone for bile salts. It's the backbone for all of our steroid hormones. So we need it in the body for us to function. Yeah. Awesome. All right. Well, now with cholesterol, so big pieces here, key takeaways. Cholesterol creation, liver cytosol, HMG-CoA reductase to get to malevolate, an energy wasting, you could say, creation or energy wasting process. We use a lot of energy to do it. The elimination pathway, primarily through bile salt formation, also in the liver, 7-alpha hydroxylase, main key regulatory enzyme, very good at recirculating or recycling these. Now I'm going to take us back to our gut and how we're actually taking things in and digesting them, because that's going to bring us into our conversation of all of these different components of cholesterol and fat that travel throughout the body. So when we are intaking fats, we're intaking them as triacylglycerols. You can see them here, triglycerides, TG. So we are taking them in, we're intaking them, and then we're putting them into our digestive tract. BS is bile salts. So we're using bile salts to essentially start breaking down our triglycerides into free fatty acids and monoacylglycerol, essentially one fatty acid molecule bound to a glycerol molecule. Those components, our fatty acids and our monoacylglycerol, is combined in a micelle. Micelle being that fatty cell that has the phospholipid bilayer, and it protects the fats on the inside. So it's going to combine these two products of our triacylglycerols into a micelle, and that's going to be taken up into the actual digestive lumen, into these cells, our intestinal cells, to be absorbed. Once it's absorbed through the bilayer, it's going to break back down into fatty acids and to monoacylglycerol. So it's going to literally break down to these products. The only reason we formed a micelle was to move it through the membrane. So we break it back down into these two components, and we reform our triacylglycerol, or essentially our triglycerides. We reform this molecule here, which is then packaged with phospholipids and ApoB protein. These are packaged all three together to create a chylomicron, which essentially is going to be the transporter of our triglycerides or triacylglycerols through the body, through the blood, or essentially through lymph back into the blood, where it will then start to circulate. And we are going to pick up on where our chylomicrons left off. So that's how we got to this chylomicron. What's in the chylomicron? Phospholipids, ApoB, and triacylglycerols or triglycerides. That's what's in our chylomicron. Okay, this kind of scary picture over here oftentimes confuses people. So I'm going to start actually with this picture here on page 73. So we started with a chylomicron all the way over on the right-hand side. And if you're a visual person, I don't have a good visualization. So this is our chylomicron. It's composed of the highest amount of tag triacylglycerols, right? What I mentioned, that 3 fat attached to a glycerol backbone. So it's the largest amount because that's really what we just absorbed. And it has maybe a little bit of dietary cholesterol, but really it's going to be a lot of those triacylglycerols. So highest tag, lowest cholesterol. Chylomicrons are going to deliver the exogenous tags, triacylglycerols, to all of your peripheral tissues. So that's kind of how we started. Okay. Now in the liver, once things get into the liver, we can see here, living in the liver, we've been creating cholesterol, we can create fatty acids, we can do all this creation of stuff. The liver can actually generate VLDL. The liver also can generate HDL. So we're going to compare these two components that the liver can generate. VLDL has also a high amount of tag, triacylglycerols, like chylomicrons, but not quite as high, and it has a lower amount of cholesterol. So large amount of fatty acids, you could say, bound to a glycerol backbone, and then some cholesterol present. This is really its purpose, VLDL, is to deliver endogenous, self-created triacylglycerols to peripheral tissues. So both chylomicrons and VLDL are delivering stuff to peripheral tissues. Chylomicrons are delivering tag to, and their source of tag is exogenous from what you eat. VLDL, its source of tag is from your own endogenous production in the liver of essentially fatty acids bound to glycerol backbone, triglycerides. Okay. VLDL then, in the tissue, VLDL can break down or generate LDL. LDL has now a low amount of triacylglycerols because we've given away a lot of these triacylglycerols to the peripheral tissues. And so now it has a lot of cholesterol. It's the highest amount of cholesterol is in this LDL molecule. This LDL molecule that came from VLDL, its purpose is to deliver cholesterol to peripheral tissues as well as back to the liver. So that's LDL's function, but primarily to peripheral tissues. And then last but not least, we have the liver and the intestine can generate HDL, primarily liver, more the intestine. And HDL has the lowest amount of fatty acids, so the lowest tag, and a high amount, not quite as high as LDL, but pretty high amount of cholesterol. And its purpose is to take cholesterol from our peripheral tissues and move it back to the liver to eliminate it or to break it down for use in other ways. This could be to generate steroid hormones. This could be to generate bile acids, bile salts, or this could be to break it down for use elsewhere. But these are kind of your main places that I want you to start, but being able to understand like chylomicrons versus VLDL, their comparisons. They both have a lot of tag, a low amount of cholesterol. Chylomicrons, their tag is from exogenous sources. VLDL, its tag is from endogenous sources. Intestine creates chylomicrons, liver creates VLDL. VLDL breaks down into LDL. There's a couple steps in between there, which we'll look at. This has the highest amount of cholesterol, and its goal is to take cholesterol to the peripheral tissues. And then HDL has a higher amount of cholesterol, and it's taking cholesterol from the tissues back to the liver. Now that we've defined these four, we'll look at the liver picture in a second, but what questions do you have about these kind of 4 lipoproteins before we get into it further?
So I have a question.
Yeah.
So I can see why having HDL is good because it's okay, good. We're taking it back for use. But then like I know this is kind of a clinical thing, but it helps me remember of like when do we get in trouble? Is it like high LDL and high VLDL, or like is one really bad and why? Or why would that get so high in a dysfunctional physiology?
So lots of reasons, right? Because we have a genetic predisposition to create more of these. So some people in familial hypercholesterolemia will see can have certain receptors upregulated or downregulated. So the body will actually increase their creation of some of these products or decrease its reuptake of some of these products. So you can see both of those in your familial hypercholesterolemia and your traditional hypercholesterolemia or hyperlipidemia. You could see endogenous sources obviously leading to a rise of TAG. And then your body has too much fatty acids. So they're creating VLDL to try to kind of combat and carry these free fatty acids around the body. So then you see LDL build up. It really depends on the source of the problem. That's really the heart. So you could see all of them build up. The one, when this lowers, it's because the body is not actually doing adequate elimination of cholesterol. And so that could be either from, I mean, most of the time, it's because the body isn't recognizing that it's in a fed state. And that could be due to insulin resistance. We can see that in diabetes. We can see that in metabolic disorder. We can also see when HDL is low in sedentary activity or sedentary lifestyle, we can see a lower HDL because the body is not in a state where it's using a lot of metabolic processes. So those metabolic processes are slower. So this number can be lower. There's a lot of different ways where this etiologies can kind of occur. We call this the bad one and this the good one because this is how we eliminate cholesterol versus this is how we deliver it to tissues for storage. So that's why LDL gets the bad rap. But really, LDL is the reason why our tissues who need cholesterol get it. So neither are bad or good. But when we talk about pathology, we typically associate this number being more dangerous because it means you're having a higher amount of fat and a higher amount of cholesterol circulating. Yeah.
Great, thanks. I think that's what I was trying to remember.
Is that kind of,
which one is the bad, the worst?
This would be considered the bad. This would be considered the good. And this is the number that we watch out for when it gets too high, where that puts you in that risk benefit or the risk group that would potentially put you in a statin benefit category, this number alone. So this is the one that we want to watch for. Yeah.
Great.
Awesome. Good questions. So I wanted to go, and then now that we've kind of seen that broken down, I like that kind of image because I think it's a little bit easier. And so I reference this page here. So if we look at the liver, we're just looking at a different pathway, and we're looking more at receptors and kind of some of these like sub proteins, these ApoC, ApoE, etc. So I'm just going to orient you to a couple structures that we already know about before we kind of go crazy here. So we already know about VLDL. We just talked about that. VLDL is produced by the liver and it goes into peripheral circulation. VLDL can be broken down eventually into LDL, right? We said that VLDL can be the source of LDL. And so how that process occurs is we see VLDL will become IDL, and then IDL can become LDL. And so that's when IDL comes into play. How do you know they're all related? They all have ApoB100 as a protein, as a part of their structure. So these three you could say are like siblings, right? Or part of the same lineage. VLDL with its ApoB100 protein is going to break down into IDL, which will break down into LDL. And then LDL will either deliver cholesterol to cells, for this instance, a macrophage, or it's going to deliver LDL back to the liver, and it can actually bind here. And then the liver can bind and retake it up and utilize it again. So this is kind of the fate of our LDL pathway, because it can go back to the liver and be either reabsorbed or reused if it's not needed elsewhere in the body. And so this process can kind of continue to go on and on. Where does HDL come into play? We'll see HDL here. I'll make it in blue so it's happier. So HDL is created by the liver, or HDL is created by the liver. HDL, though, it's showing you here, its goal is to bring cholesterol back. HDL can also essentially be kind of converted or interconvert into VLDL, or bring things back and forth between VLDL and HDL. But HDL's main goal is to take cholesterol away from something like the macrophage and bring it back to the liver, either via VLDL directly or directly binding with the liver and having the liver take the cholesterol off of HDL on its own. So that's your big processes here. Of all the proteins, I would say ApoB100 would be an important one to note. That would be my protein I would remember. And then if we remember our initial conversion of our chylomicrons here, we need our ApoB protein to even create that chylomicron. So that's the protein I would remember. Who do I have? It's the main role. So what is the main role of ApoB100? If I had to choose one main role, I'd say creation of VLDL or chylomicrons. It's involved in both their production. But I would, if I had to choose, VLDL would be the one I would choose. Does that help answer your question? Yep, awesome, great. Thanks, perfect. You probably aren't going to get asked more specific questions than that on the NPLEX, but if you want to, there are all of these different kind of apoprotein components that you could look into. I would not go further in the weeds than what we kind of already did. That's where I would stick to. Okay. All right, I want to spend a moment. Any other questions about cholesterol metabolism? Let me look at your faces. Was that helpful in at least understanding who are the players and what's their job? Thumbs up. Okay. Yeah. Great. Okay. Awesome. Thank you. That's where I think you need to be for this test. Obviously, there's always more you can know, but high yield is what we're going for. So those are the places I would lean into for this cholesterol, IDL, HDL, chylomicron, VLDL discussion. And then obviously, if you are into the biochem, go for it. Go down some rabbit holes. But I would say stick there for the purposes of the test. Okay. All right, I'm going to go back and share. And we're going to talk about two more fatty things, and then we'll probably break or we'll move into pentose phosphate pathway. It'll depend on what we're at, what time. Okay. All right. Sorry. Okay. Fatty acid synthesis. So now that we've dealt with cholesterol, we're going to start back at how does the body endogenously create fatty acids? And so why do we want to create fatty acids? Fatty acids are used all throughout the body, right? We use them in many, many different sources. We use them a whole host of different places that we need fats. So it's important that we're able to actually synthesize them. So it occurs primarily in the liver, our fat tissues, mammary glands, and kidneys. It occurs within the cytosol. It's going to actually leave the mitochondria via the citrate shuttle, again, starting with acetyl and malonyl. So these are the two kind of substrates we're talking about, but acetyl-CoA is going to be your main substrate to start with. Your key enzymatic step is acetyl-CoA carboxylase, which needs biotin as a cofactor to move forward. Fatty acid synthetase is also another enzyme. I wouldn't quite call it rate controlling, but it does have pantothenic acid, B5, as part of its structure. And your product can be any fatty acid. We use palmitate often as the example fatty acid because it's kind of it's just the common one we're going to use as our backbone. And it's upregulated by insulin. Melanocoa is a key intermediate, and it is going to use 1 ATP per melanocoa and 2 NADP per DPH per melanocoa. So to create a fatty acid synthesis, this is an energy using process. Versus when we break down fatty acids, this is an fatty acid oxidation. That's an energy generating process. So now I've shown you here. We're going to go back to our little slideshow, our resource, and we will look at our synthesis of our triacylglycerols. So there we go, fatty acid synthesis. So page 61. So to make palmitate, it's a 16-carbon unsaturated fatty acid. We're looking again, cytosol cells and liver, mammary glands, adipose, and kidney. We're going to need 8 acetyl-CoAs, 2 carbons, 14 NADPH, 7 ATP, and then we can get to our 16-carbon palmitate. 8 acetyl-CoAs we're going to essentially be able to take the acetyl-CoA and double it, right? Acetyl-CoA has 2 carbons each, so 8 times 2 carbons gives us our 16 carbon palmitate. So that's kind of how you get there. But your first big step is you have to get acetyl-CoA out of the mitochondria. So we have our acetyl-CoA is located here. To get it out of the mitochondria, I mentioned this already, but we have to convert it to citrate, and then citrate has a shuttle. So citrate can get out. Then from here, we can convert citrate back into acetyl-CoA. So that's kind of our big thing. Step 1, use the citrate shuttle to get acetyl-CoA out of the mitochondria. Step 2 is we convert acetyl-CoA into milanoil-CoA. And in this step, this is where we use acetyl-CoA carboxylase, which is the rate limiting enzyme that needs biotin. To do this, we also have to add a carbon. So we get to use some carbon dioxide, and we also have to use an energy molecule. But then we get acetyl-CoA to milanoil-CoA. So this would be your rate limiting step. If you had to learn one thing, learn this step. And then from here, we just continue to do this process over and over and over again, adding 2 carbons every time. And then once we've added 2 carbons over and over and over again, we eventually will have our 16 carbon palmitate. And so this is kind of how this process occurs. So we essentially see we're linking these milanoil-CoA units together. We created milanoil-CoA a bunch of times, and we start linking them. So we have, these are just like moving carbons. So we start from 1, 2, 3 carbons plus 2 carbons, and we get 4. And then we are going to link more, and you can see we add 2 more carbons, and then we get 6. And we add 2 more carbons, and we get 8, et cetera, until you get to palmitate. So this cycle is just going to repeat itself. So you do acetyl-CoA to milanoil-CoA, and then you link your milanoil-CoA units. Your milanoil-CoA units, you're going to link via the enzyme fatty acid synthase. And that's when you have to use NADPH. Essentially, you're reducing it, you're dehydrating it, and you're reducing it, you're moving something over, adding a milanoil, and then going again. And this cycle like repeats itself over and over. So most important pieces to note about our fatty acid synthesis: know it occurs in the cytosol. Know that you have to get acetyl-CoA out of mitochondria then via citrate shuttle. Your rate limiting step is acetyl-CoA carboxylase with biotin to milanoil-CoA. And then from milanoil-CoA, you're going to continue to essentially create these milanoil-CoA units with your acetyl-CoA substrate. Once you have your milanoil-CoA units, you're going to start linking them together using fatty acid synthase until you have however many carbons you're linking. So for palmitate, C16 is the carbons. Whatever type of fatty acid you're creating, you'll need potentially a different number of acetyl-CoAs to get to that fatty acid carbon number. But the process is the same each time. Okay.
Dr. Petshow?
Yes.
So within that process, we went through lipolysis and then lipogenesis, right? It's like a circle that you said completing.
All we went through right now is lipogenesis. We are forming fatty acids. We're going to do lipolysis next.
Oh,
Yep.
okay. This is just the formation.
This is just the formation. Yep. So within the formation, we have a cyclical nature, right? Because within the formation, we are only starting with our acetyl-CoA units. So all of these acetyl-CoA units will need to become milanoil-CoA units. And then all these milanoil-CoA units will essentially be needing to add on to this initial chain. So you'll do one acetyl-CoA to milanoil-CoA, and then you'll do another acetyl-CoA to milanoil-CoA, and then you'll link them together. And so that's kind of what this process is showing here. And then once you have your 2 milanoil-CoAs together, then you're going to take another acetyl-CoA, convert it to milanoil-CoA, and then link it to your growing carbon backbone. So you are essentially repeating both this step, step 2, and this step, step 3, over and over until you've reached your desired amount of carbons to create your fatty acid.
Okay, thank you.
Yeah, good question. Things that can regulate this process: insulin upregulates, citrate upregulates. If you have lots of citrate around, you're able to move citrate out of the shuttle and convert it into acetyl-CoA. Things that are going to decrease this is glucagon or palmitoyl-CoA if you have a lot of palmitate or if you have a lot of your product. Okay. Now, we're not going to talk about elongation or saturation. We're going to talk about breakdown. So fatty acid oxidation. Let me find my oxidation. There we go. Beta oxidation. So I want, yeah. So we talked about lipogenesis here, and I'm just going to make sure, yeah, this is fatty acid metabolism. Perfect. So now we're going to talk about lipolysis, the breakdown of our fatty acids or our fats. Our fats are going to travel in our blood, either from the gut or fat tissue, and they're attached to typically a protein like albumin. They're going to get into mitochondria via carnitine, the carnitine shuttle. So that's how fats will get inside our mitochondria again. So we now have our fatty acetyl-CoA inside of our mitochondrial matrix, and now we can undergo beta oxidation. How do we get carnitine from lysine and methionine? So that is kind of an important, that could be an amino acid component that you could be asked. And vitamin C, it's questionable if we need it for synthesis or not. So I wouldn't care about that. I would just focus on carnitine, lysine, methionine. So beta oxidation. Once we have our fatty acetyl-CoA inside our mitochondrial matrix, so our fatty acid, we can now break it down. This is called beta oxidation, and this process also, we'll kind of see there'll be multiple steps that we're going to undergo. Our main rate limiting enzyme is none of these things here. It's actually our carnitine acetyltransferase to get our, let me find it, carnitine, carnitine acetyltransferase. I might not even have it on here. Well, that's unfortunate. Okay, well, let me pull it up. That will be helpful for us to see. Okay, no, just kidding. Sorry, y'all. Carnitine acetyltransferase can get you into here. Our rate limiting step, we'll pull this one up. This will be easier. Is going to be here. Acetyl-CoA synthase. Key enzymatic step for beta oxidation breakdown. So, no, that's totally wrong. Okay, ignore this. Ha, hilarious. Sorry, y'all. I love when my slide set isn't updated. Okay, we'll get to here. I got us. Okay. So our major, I'm just pulling up my correct notes. And we will get to our page. Okay. Great. Yes. Okay. So our major rate limiting enzyme, and I will update this on the notes so that we all have it here. So our fatty acid is outside here. We have to first combine it with our CoA molecule. This is going to use energy, and it's going to use fatty acetyl-CoA synthetase. Our next kind of rate limiting step is actually getting this through the carnitine shuttle. That would be your rate limiting process, is this whole carnitine shuttle here as a whole, all of these enzymes involved. This is why it didn't match my picture, is carnitine acetyltransferase 1, or this essentially carnitine palmitoyltransferase 1, the CPT1. This is also referred to carnitine acetyltransferase 1. So this molecule here, moving our fatty acid CoA molecule from the cytosol into essentially into the mitochondria and the outer mitochondrial membrane, is the first rate limiting step of this process. From there, we have our transferase 2 that takes us from the outer membrane into the inner membrane. If we can get this into the inner membrane, this process will likely move forward because it is going to be an energy generating process to actually break down fatty acids into energy, because what we'll end up with is acetyl-CoA at the end of the day from beta oxidation. So your two steps that are important to remember is fatty acids plus CoA gets you fatty acyl-CoA, and that's via acyl-CoA synthase. That's going to occur in your cytosol. From there, it's going to be your transfer through our mitochondrial membranes. So our fatty acyl-CoA to get from one side to another, which is essentially your carnitine transferase molecules. You have one to get you through the outer membrane and two to get you through the inner membrane. Sorry about that, y'all, but I wanted to make sure we had it right. Inside then you have fatty acyl-CoA and carnitine. Carnitine can then be shunted back through and utilized again to help this process continue to move forward. That's the carnitine shuttle. Once we have our fatty acyl-CoA inside of our mitochondrial matrix, then we can undergo beta oxidation. You'll see through this process, you're going to actually generate ATP as we move down. So you'll have fatty acyl-CoA is going to generate 1.5 ATP because you get an FADH2 from your first step, your acyl-CoA dehydrogenase. Then your third step, you get an NADH through beta hydroxyacyl-CoA dehydrogenase. All I would remember here is I just remember your dehydrogenase enzymes in beta oxidation are the ones that generate energy for you. They're the ones that are going to give you ATP. At the end, you're left with, when you do this process, you cleave off acetyl-CoAs. And so you're going to continue to do this process however many times you need to if you have a carbon structure that's 12, you're going to undergo this process 5 times because you're going to break them apart in carbons of 2. And so you'll end up with 6 acetyl-CoA molecules if you have 12 carbons that you're starting with, if that makes sense. You're essentially undergoing an oxidation, a hydration, so where we add water, an oxidation, and a cleavage. That's what your processes are here. So we're oxidating, we get energy. We're hydrating with water, essentially making it less stable, more sterically reactive. Then you're oxidating as well, again, getting energy, and then you're going to cleave here, essentially bumping off these carbon molecules at the end. So you are left with this bond being broken here. So you get your acetyl-CoA, your 2-carbon acetyl-CoA. That's your process of beta oxidation. And so this is also cyclical. So as you're mentioning before, both processes, fatty acid synthesis, so lipogenesis and lipolysis, are both cyclical activities. That would be appropriate. Jocelyn, yes. Awesome, great. All right, I know that was a lot. So it's giving you an example of how we can get ATP generation. So you can see breaking down of a fatty acid can generate way more ATP than breaking down glucose. So fatty acids gives you way more energy generation. That's why it's such a high yield molecule if we can use it for energy. There's a slightly different channel for breaking down odd chain fats. I would not even worry about that for the purposes of this study. Okay. All right. Any fat questions before we kind of get into we'll do a 10-minute break, and then we'll do our last discussion. Any fat questions before we kind of move into this last section? Okay. Have you been filling out your table? I hope you have. Don't forget about it. You're going to be your big study piece. That would be the one thing I'd focus on learning in the next week and a half, would be that table. You can learn that table before boards at this point for biochem. That's going to be your most high yield component. Okay. All right.
Table.
Sorry.
You have for beta oxidation, you have acetyl-CoA synthase as the key enzymatic step. Would you also put the CPT1 and 2 on there?
I would say that acetyl-CoA synthase is the rate limiting enzyme for the actual conversion, but the conversion can't take place if you don't get it through the actual carnitine shuttle. So I'd keep that as the rate limiting enzyme, but sometimes, when I have 2, my next would be that carnitine transferase. So if that makes sense. Yeah. Excellent. But if you had to learn just one, I would learn your first one, fatty acyl CoA synthetase. Great. Awesome. Perfect. All right. I'm going to have us break. We'll come back at 1 minute after 11. So we'll do a 10-minute break. And then we're going to go through pentose phosphate shuttle. We're going to go through ketogenesis and ketone degradation. And then we'll see what we're left with time to cover any kind of other components for the rest of the hour. Thanks, y'all.